Welcome back to the Deep Dive. Today we're digging into a really foundational paper from 2013, the one that introduced ATAC-seq.
That's right. And the core idea, maybe the most surprising thing, is how it took these really complex multi-day methods.
Like DNase-seq, FAIRE-seq.
Exactly.
Yeah.
Methods that needed tons of starting material, millions of cells sometimes. And ATAC-seq replaced them with this, well, incredibly simple two-step protocol.
Simple, same day. And it works with, what, like 500 cells? Which is just wild.
Right. And on top of that, you get 3 different kinds of epigenomic data out of the same experiment: accessibility, nucleosomes, and even transcription factor binding.
It really addressed a huge need. Before ATAC-seq, trying to map open chromatin, the accessible regulatory bits of the genome, was tough.
It really was. Those older methods, like you mentioned, DNase-seq, FAIRE-seq, they needed so many cells, often millions.
Millions. So forget about using small patient biopsies or rare cell types from primary samples then.
Pretty much, yeah. Or at least it was extremely difficult. And the protocols themselves were long, lots of steps, lots of chances for things to go wrong or lose your sample.
Okay, so the authors saw this problem. They needed something fast, sensitive, low input, and ideally integrated, giving you the whole picture at once.
Exactly. They wanted accessibility, nucleosome positions, and TF occupancy, all from one go.
And their big idea involved this bacterial enzyme, right? Hyperactive TN5 transposase.
Yeah, TN5, it naturally cuts DNA and sticks in other little bits of DNA. So the question they asked, sort of the core of Figure 1, was, could we load this TN5 thing up with sequencing adapters and just throw it at native nuclei, unfixed cells? Would it selectively cut and paste those adapters only into the open, accessible chromatin regions?
And spoiler alert.
It worked beautifully. The whole assay is basically just 2 steps. Add the loaded TN5 to the nuclei, let it tagment, and then just PCR amplify everything.
Simple as that.
Simple as that. And Figure 1 showed it worked incredibly well. They got great accessibility maps from 50,000 cells, sure, but also all the way down to only 500 cells.
500, that's the number that just jumps out. How did the signal quality hold up compared to, say, DNase-seq needing millions of cells?
That was the amazing part. The signal to noise was actually very similar, despite using, 3 to 5 orders of magnitude fewer cells.
Wow. So how's that possible? Just way more efficient.
It's because the TN5 is so specific for open chromatin, and the whole process is so streamlined, you avoid almost all those lossy cleanup steps from the older methods. It happens mostly in one tube.
Okay, so that covers the accessibility mapping, the first piece of data, but you said 3 types.
Right, so this is where Figure 2 comes in, and it's really clever. They realized the size of the DNA fragments they were sequencing wasn't just random.
It actually meant something.
It contained information about nucleosome positioning. When they plotted the distribution of fragment lengths, they saw this really clear repeating pattern, a peak about every 200 base pairs or so.
Ah, the length of DNA wrapped around a histone core plus the linker.
Exactly. It perfectly captured the regular spacing of mononucleosomes, dinucleosomes, and so on. So, boom, accessibility and nucleosome architecture from the same sequencing library.
Okay, that's 2 down. Accessibility from where it cuts, nucleosomes from the length of the fragments. So here's a question for you listening. If fragment lengths tells us about nucleosome packing, what other kind of information might be hiding in the sequences between those cuts, especially where proteins might be bound protecting the DNA?
Good question to pause on.
What did they find?
They found the third piece, transcription factor footprints. This is mainly in Figure 4 and also Figure 3.
Footprinting, so where a protein is sitting, the TN5 can't cut.
Precisely. If a transcription factor, like the classic example CTCF, is bound tightly to its specific DNA sequence, the TN5 can't access that little stretch. When you map all the reads, you see a dip, a footprint, right at the known binding site for that factor.
So you can infer TF binding activity. And they connected this back to the nucleosomes too.
Yeah, that was Figure 3E. They looked at where these TF footprints occurred relative to the nearest nucleosome, and they found different patterns. Some TFs, like CFOS, seem to really avoid nucleosomes altogether.
Okay.
Others, like CTCF again, tend to bind right up against the edge of a nucleosome, almost nestling next to it. And some seem to bind right over where the nucleosome DNA would be. It gives you this integrated view of TFs and chromatin structure.
It's just elegant, taking multiple complex assays and collapsing them into one enzymatic step.
It really was revolutionary. And the implications, especially for clinical work, were immediate.
Right. Figure 5 gets into that. They actually timed it, didn't they? From taking a blood sample, isolating T cells, doing the ATAC-seq prep.
All the way to being ready for sequencing, under 275 minutes, less than 5 hours.
That's fast. fast enough to potentially influence decisions for a patient relatively quickly. Personal epigenomics in near real time.
Exactly. And they showed a direct example. They looked at T cells from someone, focused on the IL-2 gene locus, which is important for T cell function. Using the footprinting data, they could see that a key signaling protein, NFET, was clearly bound and active there. But other potential drug targets, like IRF4 and STAT3, weren't showing footprints.
So you could actually see which pathways were active in that person's cells right then, helping you choose maybe which drug might be most effective.
That's a potential, yeah. Rational drug targeting based on the actual epigenomic state.
Okay, let's wrap this up. If you take away 3 things about ATAC-seq in this paper, what should they be?
First, I'd say the method itself, using TN5 transposase for the simple, super fast 2-step protocol.
Second, the sensitivity. It works reliably with incredibly low cell numbers, down to 500 or even fewer now.
And third, the integrated data. You get accessibility, nucleosome positions, and TF footprints all from a single sample, a single library prep.
Fantastic summary. And maybe a final thought to leave everyone with.
Well, the authors themselves pointed this out back in 2013, and it's probably even more true today. They predicted that the bioinformatic analysis making sense of all this rich data would become the main bottleneck in epigenomics, more so than actually generating the data.
So, thinking about the future,
Hmm.
what does that shift mean for the skills that biologists need moving forward? Something to chew on.
Definitely something to think about.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has 3 main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the 3rd goal. Let's go! Let's make great science together.